So first of all, thank you, Davor, for the invitation to speak today. I think the ISHR JMCC webinar series has been a tremendous success. It's a huge undertaking to have a webinar every day from really great people all around the world. And while I like looking at an audience, I'm getting used to Zooming and sharing a work this way. So what I want to talk about is work that we've done over the last, 10 or 15 years relating to the role of oxidative stress in the brain and its role in sympathetic activation in heart failure. So let me, whoops, there's my pointer. So I don't have to tell anybody that's listening in, that's watching this, that heart failure is a huge issue in the United States. It's estimated that there'll be a 46% increase in people living with heart failure in 10 years from now. And of course, it's a huge economic burden. And the etiology of heart failure is various, a lot from ischemic and much of it from dilated cardiomyopathy of other causes. The problem with heart failure is that it is a positive feedback mechanism. Certainly the adaptive mechanisms that occur following a cardiac insult are compensatory. Once there's a drop in cardiac output, the system upregulates neurohumoral activation, which includes both the sympathetic nervous system and the renin-angiotensin system. Unfortunately, in the short run, this is good, but unfortunately, chronically, both of these things feed back onto the peripheral circulation and the heart to make the heart failure worse, result in more myocardial cell death, more remodeling occurs. There are a variety of electrophysiological abnormalities, and there's this reverberating vicious circle that ends in death. Now, what our interest has been is the mechanisms that are responsible for activating sympathetic outflow in the brain. And we have focused on several areas in the brain, mostly in the brain stem. Others in our group have focused on the hypothalamus, but most of what I will talk about are neurons in what we call the rostroventrolateral medulla. So here's kind of a roadmap of what I'd like to cover today. Talk a little bit about sympathetic activation in heart failure, and then move into oxidative stress in general. Most of the talk will relate to this interesting multi-dimensional transcription factor, NRF2, that is expressed throughout the body, but as we'll see, expressed in the brain as well, especially in presympathetic neurons. And then finally, some new work that we have done on the regulation of NRF2 in heart failure through microRNAs, exosomal transport, and crosstalk between tissues. So this just is a slide that I like to show when I give these talks because it really documents the massive increase in sympathetic outflow that occurs in heart failure. On the left are old data from Jay Cohn showing plasma norepinephrine and the probability of survival over a 5-year period stratified to plasma norepinephrine. And you can see that those patients with the highest levels of plasma norepinephrine have the poorest survival profile. And of course, Mike Bristow has shown many years ago that increases in sympathetic tone result in downregulation of beta-adrenergic signaling in the heart. And this has become well known. If one records sympathetic nerve activity using the technique of microneurography, you can see that, and this is muscle sympathetic nerve activity, you can see as cardiac index falls, there's a progressive increase in sympathetic outflow. So I think there's very little question now that, that part of the pathogenesis of high levels of sympathetic outflow is mediated by central mechanisms. So in our lab, we use several different models of heart failure. Probably the most common is the coronary artery ligation model in the mouse and rat. And this just shows you what that looks like. I'm sure many of you are familiar with this. Liang Zhao, when he was in the lab some years ago, he's now at David Harrison's lab at Vanderbilt, made these measurements, and he showed very clearly that if you record electrophysiological activity from the renal sympathetic nerve in animals with chronic heart failure, there's a marked increase. And this is very similar to what is shown for muscle sympathetic nerve activity in humans. We have a rabbit model of chronic pacing where we pace the ventricle in the rabbit for several weeks at rates approaching about 400 beats a minute. And when we look at renal sympathetic nerve activity, in this case, in the conscious animal, you can see a similar increase. So we got interested in the whole idea that neuronal activity in the brain, as has been shown in the periphery, is modulated by high levels of oxidative stress. And it had been known for a while that in heart failure and in hypertension and many other disorders, there's abnormally high levels of oxidative stress, not only superoxide, but high levels of hydrogen peroxide and other oxygen radicals. And the derivation of these free radicals are from a variety of sources, but the two most that have been really investigated are the source from membrane-associated NOxes, in this case NOx2, and from a mitochondrial electron transport chain with the leak of superoxide. There are many other ways oxidative stress can be made in a variety of tissues, including uncoupling eNOS. And we got to thinking that it's really important to understand this because we know that oxidative stress can modify ion channel function and alter action potential generation in many tissues, but in the central nervous system especially. So high levels of active oxidant species can result in increase in neuronal activity by downregulating potassium channel activity or upregulating calcium channel activity by modifying channel proteins directly. So as I said, we were interested in a part of the brain that houses the presympathetic neurons in the final common pathway, and that area is the rostroventrolateral medulla. Here's a schematic, and you can see here that it's a very small area, but a very potent and complicated area with projections from many other areas of the central nervous system. And this just shows that input to the rostroventrolateral medulla comes from other medullary sources, from peripheral inhibitory mechanisms to modulate outflow. And there are projections, many of them glutamatergic, to the spinal cord for final outflow to the heart, arterioles, kidneys, and every other tissue. There's input from the paraventricular nucleus in the hypothalamus, and probably even cortical descending pathways that modulate output. So one of the early experiments that we did looking at oxidative stress in this area of the brain was done in collaboration with Li Gao, a close associate of mine who've worked in the lab for many years. And we used this rabbit model and paced the animals until their ejection fractions were 40% or below. So this is a non-ischemic dilated cardiomyopathy. And when we looked at markers of oxidative stress in the rostroventrolateral medulla using dihydroethidium staining and lucigenin, NADPH dependent lucigenin, we found much higher levels in animals with heart failure compared to sham control animals. Matt Zimmerman and Paul Fidel did a study in humans that had stable but chronic heart failure, and they looked at oxidative stress using electroparamagnetic spin resonance spectroscopy. And you can see, just as we see in our animals, now this is peripheral blood, in PMNs, there's high levels of oxidative stress, those total ROS and superoxide in those patients with chronic heart failure. If we look at one of the more ubiquitous types of antioxidant proteins expressed both in mitochondria and in the cytosol, MNSOD and copper zinc SOD, we find that, that it's decreased in heart failure. Don't pay any attention to the exercise paramine, that's a whole nother seminar, but exercise can modulate this. And we see that for both isoforms of superoxide dismutase. Furthermore, if we try to overexpress SOD and in this case, it was not targeted to the RVLM, but it was given a virus that overexpresses copper zinc SOD was given into the cerebral ventricles. You can see that this high level of sympathetic nerve activity that is characteristic of heart failure can be progressively reduced in a dose-dependent manner when we overexpress SOD. Matt and graduate student Aaron Rosenbaugh did a study some years ago in which they administered a nanoparticle, which is a polyethylamine pegylated polymer containing copper zinc SOD. And they gave this to rabbits with heart failure, or actually they gave it, in this case, to normal rabbits, and then challenged the rabbits with intracerebroventricular injection of various doses of angiotensin, which, as most of you are aware, is a really great activator of NADPH oxidase and generation of superoxide. And it was shown that with this nanoparticle, you can get a dose-dependent reduction in the change of arterial pressure mediated by angiotensin. And this increase in arterial pressure is mostly sympathetically mediated. And we saw no effect with just empty polymer or with naked protein by itself, because it was metabolized. But when we gave the long-lasting, what we call a nanozyme, nanoparticle, there was a dramatic reduction in the pressure response elicited by central angiotensin. Okay, so as has been clear for a long time, the balance between antioxidant elaboration and oxidant stress is what is the final determinant of strain on a system. And if oxidant stress outweighs the capacity to scavenge these antioxidants, the result is a pathology, which is in many forms. There is inflammation, DNA damage, a variety of protein modifications, and tissue injury that in some cases is irreversible. So that got us to thinking about how antioxidants are regulated. And I want to turn our attention now to a discussion of this transcription factor, NRF2, which is a basic leucine zipper protein that is considered to be one of the master regulators of antioxidant protein synthesis. So NRF2 is located in two places. In the cytosol, in conjunction with its tether Keep1, so this is an endogenous inhibitor of NRF2. Keep1 has been well established to be an oxidant sensor. It has a variety of cysteine residues, and there are 3 primary cysteine residues with sulfhydryl groups that can be oxidized. Once those sulfhydryl groups are oxidized, there's a conformational change in Keep1 and a release of NRF2 that translocates into the nucleus. It's phosphorylated primarily by protein kinase C, and then translocates into the nuclease, where it binds together with a variety of other small transcription factors to the antioxidant response elements on DNA that mediate the downstream transcription of a variety, and this is just a very, very short list here, a variety of antioxidant proteins. If there's low levels of oxidant stress, NRF2 and KEAP1 stay together and eventually are ubiquitinated and then degraded in the proteasome. So this whole E3 ligase, ubiquitin E3 ligase complex, including culin-3, is very important for determining the levels of NRF2 and whether or not there's enough NRF2 to translocate to upregulate antioxidant proteins. The regulation of NRF2 itself has been studied for several years now, and in addition to its turnover by the E3 ubiquitin ligase system, its translocation with other small regulators, there is a lot of work that has been done on transcriptional regulation. Interestingly enough, NRF2 can actually bind to its own gene sequence and upregulate its own transcription. And then there's a variety of modulation, including microRNAs, in the postscriptional setting that I want to talk about in a little bit. So one of the reasons NRF2 has become so popular is that it's found its way into the lay press. There are a variety of natural, for lack of a better word, nutraceuticals that have been marketed that apparently are actually very good NRF2 activators. The one that you may be familiar with is curcumin, which is the active ingredient in the spice turmeric. As it turns out, there are several clinical trials using curcumin now. You can buy it certainly over the counter, but in terms of real science, there are a few studies now that are looking at the validity and the usefulness of curcumin in many diseases as NRF2 activators. There's a company called Life Advantage that was started by Joe McCord, who is one of the co-discoverers of SOD, which is there's a product called Protandim, which is touted to be a NRF2 synergizer. This is all over the counter, and if you go to the literature and you read about this, you'll find all sorts of claims. Some of it may be somewhat valid, but I think there is not enough clinical studies at doses needed to activate NRF2 to see if this is really beneficial in disease states. So the first question we asked is NRF2 in the brain associated with sympathetic nerve activity and heart failure? And does that affect outcomes? The idea was that any insult to the myocardium would activate excitatory reflexes and maybe reduce inhibitory reflexes for an initial increase in sympathetic outflow to both the heart and the peripheral circulation. And as I said, this is a short-term modulator. What happens chronically is maybe mediated by molecular changes in many areas of the brainstem, including the medulla. A postdoc in the lab in rats looked at the expression of NRF2 in the RVLM of rats with coronary artery ligation heart failure, and a graduate student, Ahmed Wafi, who's just about finishing up his PhD now, has looked at this in a mouse model. And one of the things that we noted in both models is that if you look over a variety of physiological and pathological states, we find that there's a correlation between ejection fraction, or cardiac function, if you will, and normalized NRF2 expression in the rostroventral medulla, both for protein and for message. And so that got us to thinking that, wow, maybe there is a real regulatory mechanism here with some sort of feedback from either local oxidative stress in the brain, mediated by metabolic activity of those neurons, or communication between the periphery and the central nervous system. So the first question we asked is, what happens if we knock down NRF2 in the rostroventral medulla? Does it increase oxidative stress? And if it does, what happens to sympathetic outflow, cardiovascular reflex function, in this case the baroreflex, and arterial pressure? So we worked together with Li Gao again, who's been a real mainstay in our laboratory, and Shyam Biswal from Johns Hopkins, who at the time had a variety of models, and one of them were NRF2 flox mice. So we obtained these NRF2 flox mice some years ago, and we bred them up in our laboratory. And then we delivered a viral crete into the RVLM using stereotaxic coordinates. This was done in these mice, and then over time, we looked at a variety of parameters. So the idea here would be if you knock down the NRF2 Keep1 complex, we'd see increases in ROS, and that would result in increases in sympathetic outflow and perhaps deleterious effects to cardiac function, but certainly should lead to a change in blood pressure. So this just shows some of the results from that study. We were able to knock down NRF2 message by about 55 or 60%. And you can see that NRF2 protein was very low. Remember, we actually expected to see very low levels even in our GFP, but there is certainly levels that you can see, especially if one uses a good monoclonal antibody for detection in Westerns. For immunofluorescence, when we delivered the lengthy GFP, what we found was that in the area of the RVLM where the GFP was expressed, there was no NRF2 expressed, and you can see the overlapped image here. So we were pretty confident that we could deliver our virus to a very discrete area in the RVLM and knock down NRF2 so we could look at the outcome of that manipulation. When we did this, we looked at a few antioxidant enzymes, and Cre delivery decreased NQO1, HO1, SOD2, and catalase. Clearly, it doesn't knock down these proteins by 100%, and that's probably because when we punch out these areas of the brain, especially in mice, we get peripheral tissue that is not affected by the virus. So I think we were pretty happy that we got the kind of decreases in both NRF2 and antioxidant enzyme expression that we saw. We also looked at some markers of oxidative stress following the lenticree injection, and this just shows that under normal conditions, when we just transfect with GFP, we don't see much DHE staining, but after Cre transfection, many neurons have a nuclear expression of dihydromethidium. Matt Zimmerman, who runs our EPR core, tried to analyze punches of the RVLM using EPR, and this is difficult to do because the amount of tissue we have is very low. We had to pool animals, but we did find a reduction in the signal an increase in the signal when we transfected with Cre, suggesting that this is probably most likely due to more superoxide. Then we put telemetry units into these mice to record blood pressure on a chronic basis. And in our GFP transected mice, you can see there's a nice diurnal variability in blood pressure. These are conscious measurements now. And you can see blood pressure increases during the nighttime hours and then decrease in the daytime. In our animals with Cre administration, there was about a 20 to 25 millimeter increase in arterial pressure. This really kind of surprised us. because I would have thought that the level of NRF2 in normal animals would be relatively low if they weren't under some oxidant stress, and that we wouldn't see much change by knocking it down. But indeed, there's enough there apparently to result in a restraint of sympathetic outflow. We didn't see much change in heart rate, however. We looked at indices of sympathetic nerve activity. We showed that norepinephrine was markedly elevated in norepinephrine excretion. And when we looked at direct recordings of sympathetic nerve activity, you can see that there's marked increase in renal sympathetic nerve activity in animals with heart failure. And following CRE administration, that is decreased, and you can see, is increased markedly. And what's more prominent is that if you try to suppress sympathetic nerve activity by increasing arterial pressure, it's usually in the GFP animals completely silenced, whereas in CRE animals it almost doesn't turn off at all. So to summarize this group of experiments, we think that NRF2 might be very important in sympathoregulatory areas like the RVLM. When we knock it down, we get increases in reactive oxidant species. We get decreases in antioxidant enzymes, resulting in sympatho-excitation and hypertension. So what about the other side of the coin? What happens if we upregulate NRF2, if we overexpress it in the RVLM? Again, Li Gao and a graduate student who just finished up, Anyun Ma, did these experiments. And what we relied on here is a virus that we got from David Patterson at Oxford, who gave us a lentivirus that that was driven by both the tyrosine hydroxylase and the CAMP kinase 2A promoters. CAMP kinase 2A is a promoter that is found primarily in glutamatergic neurons. We took out, his virus had a NOS gene in it. We took that out and replaced it with NRF2. And we used a construct in which we mostly gave the CAMP kinase 2 virus to the RVLM, or we used Keep1 flox mice and Knockdown Keep1 to upregulate NRF2 by CRE administration. So this just shows some data from the first set of experiments in which we overexpressed NRF2. And to make a long story short, in heart failure animals, when we overexpress NRF2, as you can see here, it turns out that when we do this, we overexpress it both in sham and heart failure animals. But interestingly, we get increases in NQ01, we get increases in HO1, we show nice overexpression of NRF2, and we suppress sympathetic nerve activity. I won't go through all of the hemodynamics in this model. In spite of the fact that norepinephrine and sympathetic nerve activity are lower, cardiac function really doesn't change very much. And I subscribe this to the fact that in this model, we have a massive infarct that is probably 50 or 60% of the left ventricle, and it's very hard to see recovery from that. This is another a marker. Here you see sympathetic nerve activity in our heart failure animals, very high, and after overexpression of NRF2, baseline is lower, and barrel reflex silencing is restored, and you can see the mean data described here, and these are just baroreflex mediated parameters that I won't go into in great detail. The other option here is to knock down Keep1, which we did in Keep1 floxed mice, also from Shyam Biswal at Hopkins, and the same results. So here the strategy is to knock out the Keep1 complex, increase the amount of NRF2 translocating to the nucleus, and this is immunohistochemical staining, showing that we can upregulate NRF2 when we knock down Keep1. And again, we see, when we do this, we see suppression of sympathetic nerve activity, decreases, I'm sorry, when we upregulate NRF2, we see decreases in sympathetic nerve activity, both, and here's the actual neurophysiology and the recording, as well as norepinephrine. So this study kind of suggested that upregulation of NRF2, and this can be done, by the way, with NRF2 activators like curcumin and sulforaphane and dimethyl fumarate as well. But this strategy showed, I think, strong proof of principle that NRF2 it plays a role in sympatho-excitation in the RVLM in the setting of chronic heart failure. So the next topic I want to turn to as we kind of finish up here is whether or not there's some communication between the heart and the brain. We know there are lots of neural communications, many reflexes, but Shanghai Tian came up with the idea that there may be regulation of NRF2 translation through delivery of microRNAs via the extracellular vesicular pathway through the circulation. One of the things we noticed, and now I'm going back to our infarcted mouse hearts, and you can see that what we noticed that if you look at NRF2 MRA in the normal myocardium, you find that it's upregulated. mRNA is upregulated. But interestingly, protein is significantly downregulated. So that got us to think that, and of course the downstream antioxidant messages are also downregulated, and two markers of oxidative stress in the heart, 4-hydroxynonenal and 8-hydroxydeoxyguanosine, were also upregulated. So that got us to think that there must be a regulatory mechanism reducing the translation of this increased mRNA signal, and that's why we turn to an investigation of specific microRNAs. And when one searches the literature, you can find specific microRNAs from many tissues in which NRF2 mRNA is a target. In humans, this has been, there's been a variety of papers over the years. This is an older paper from Gerald Doran's work showing that tissue levels of microRNAs from patients that have gone on to LVAD assist devices with failing hearts showed marked changes in microRNA profiling. And one of the microRNAs, miR27A, is one that we have been very interested in because it does have NRF2 as a downstream target. We obtained a variety, we have a very large LVAD program here and a tissue bank with now close to 400 samples. We do about 75 LVADs a year here and about 50 or 60 transplants. So we have lots of left ventricular cores, and we took advantage of this tissue. We don't have so many donors, but these are hearts that were harvested but were never used for transplantation, but are otherwise normal. And what we found when we looked at the microRNA profiling in these LVAD hearts is that there were several microRNAs that were upregulated. There were a 34A, the passenger strand, 142 5P, 144, and 153, and 27A was upregulated as well, just did not reach significance, and we could not find a change in 28A. So this was done in collaboration with Dan Anderson, who's our chief of cardiology here. So going back to the infarcted mouse model now, we looked there again. and we looked at the microRNAs, and we found again that these same microRNAs, 27A, 28-3P, 34A, were upregulated. Other microRNAs, which also have been shown in the MIR database to have NRF2 as a target, were not different. So one of the things Dr. Tian did is he looked to see if we could mimic changes in NRF2 in tissue culture. So he took primary cardiomyocytes, incubated them with microRNA mimics, and found that, and then he stimulated these cells with TNF-alpha, which increases oxidative stress and translocates NRF2 very well. And we found that NRF2 expression increased with TNF-alpha in non-transected cells or in cells with scrambled microRNA, but that was totally suppressed when MIR 34A, 28, and 27 were administered. If we go back to the brain now, into the RVLM, we find that these same microRNAs are elevated in the heart failure animals. So 28, 27, and 34, and there's, of course, as I showed you before, increases in oxidative stress. So now the question is it possible that a message gets from the heart to the brain to modulate NRF2 oxidative stress and sympathetic outflow? So we turned to the whole idea that there were microvesicles that were released during myocardial damage that was capable of sending a message to other tissues to regulate NRF2. So we isolated microvesicles and we analyzed the cargo from these microvesicles in cultured cardiac fibroblasts. At least we started with cultured cardiac fibroblasts. And we characterized these to be a variety of sizes, but we had a strong peak at about 80 nanometers, suggesting that these are in large part exosomes. When we stimulated with TNF-alpha, sure enough, we get big increases in 27A, in 28A, and in 34A. We don't see these, this is in the media now. In the tissue, in the myocytes themselves, we don't see much change. So that suggests that these exosomes are stimulated by TNF-alpha, released rapidly, and don't really increase very much in these cardiomyocytes. And then what we did is we co-cultured rat fibroblasts that we labeled with green fluorescent protein with cardiomyocytes, what we labeled with M-cherry. We co-cultured them, stimulated them with angiotensin, a good oxidative stress, and then looked for green fluorescence in the myocytes. And this is what we saw. These are the myocytes in the bottom of the well, and you can see that there are green particles together with the red particles, and what we see here is overlap largely around the perinuclear zone. And this is a little video. In this case, exosomes were labeled with red, and there was a lexofluorophthalotoxin that labeled the actin myofilaments with green, and you can see that the exosomes do get taken up and are in the cytosol. What about in the circulation? So in the circulation, we also looked at that in our heart failure model. And here again, we see miR27A much higher in animals with heart failure in plasma, at 28A and 34 similarly increased. Again, we characterized these by both electron microscopy and nanosite light scattering. So, the idea here is that we can get cell-to-cell communication. We think we can get release into the circulation, and we can get transport of these extracellular vesicles with their cargo, which we believe contains microRNAs that may target NRF2 and modulate oxidative stress. One experiment that is kind of exciting, and this is still very preliminary, but I wanted to show you here, is we took microvesicles, we characterized them, labeled them with PKH26, which is a red fluorescence. We injected them into recipient mice, and then we subjected them to IVUS imaging at 4 hours and 24 hours, and we looked at expression in many of the organs. And what we found was that there was uptake by some of the organs, but the brain, especially at 24 hours, seemed to concentrate these exosomes. And we thought, well, maybe they're just sticking to cerebral blood vessels. But when we sectioned the brain and we looked at specific neurons, you can see exosomes are taken up into these isolated neurons in the RVLM, from the RVLM, and again, seem to be distributed in close to the nucleus. And you can see the particles here in the slices through the RVLM. The brain extracellular vesicles contain, the cargo contains both 27A, 28, 34, and a couple of other microRNAs that are thought to be cardiac specific. That's why it was so surprising to us that we found increases in microRNA 1, 208A, and 499. So if you look at the literature, these three microRNAs tend to be cardiac specific. So that suggested that maybe they are getting to the brain from the heart. And we then did what is still a very preliminary experiment, but kind of exciting, in which we try to transfer the phenotype from heart failure animals to normal animals by injecting extracellular vesicles harvested from heart failure animals, transfected with antigomeres to these specific RNAs, labeled and injected into the RVLM. And right now we only have one marker, and that is plasma norepinephrine. This is an N of 2, so don't take it, please take it with a grain of salt. But what we find is that with just heart failure extracellular vesicles, there's an increase in norepinephrine excretion, and when we co-transfect with antigomeres to these specific microRNAs, that is normalized. And you can see here that definitely microvesicles are getting into the area of interest. And NRF2 is also upregulated when we incubate with the antigomeres. And just finally, very quick, we're currently using a red-green reporter mouse that is crossed with a myosin heavy chain, tamoxifen-driven, so we can track green particles from the heart to other tissues. And so when we take these mice and we treat with tamoxifen, particles from the heart, this shows that the heart expresses, this is treated with tamoxifen, the heart is, all myocytes are red, non-myocytes, as shown here, myocytes are green, non-myocytes are red, and when you merge them, you see here, and when we look in the brain, we can see green particles in the brainstem, suggesting that these must have been derived from the heart. So we're using this model now as a way of providing evidence that there is this non-neuronal microvesicular communication between the heart and the brain. So this just summarizes the kind of the overall story. I'll skip this, and NRF2 activators may prove very useful in treatment. There are several clinical trials now using NRF2 activators for a variety of diseases. And I just want to end by thanking many people that have worked for many years, whose work I showed, some of whom, like Julia Shanks, who's in New Zealand right now, came from David Patterson's lab, who had worked on the stellate ganglion and NRF2, which I didn't show. So I thank you very much, and we'll take some questions.
Okay.
So,
thank you, Irv. Great story.
okay. So, do you want me to just read through these questions? Is that
So thanks for that, Irv. Yeah, so I think if, as you've seen on the other webinars, if you go through them and we'll see how far you get. I think there's about 10 of them or so. So you should be able to get through those, I guess.
Just bear in mind the questions tend to come in as you start answering them. So,
okay, well, I'll go through the Q&A. Looks like we have a couple chat things as well. But so Hector Chapoy sends a question: is what do you think about the use of cannabidiol like anti-ROS in heart failure, activating to NRF2? I don't know. I mean, nothing surprises me anymore, and certainly cannabidiol has been used for a variety of disorders. I think it has been shown to have an antioxidant property. Whether it works through NRF2 or not is completely unknown, at least not that I'm aware. There are some studies that have shown that this drug may be useful in inflammation, and there is a very important a regulatory mechanism between inflammation, NRF2, and NF-kappa B, and so I think that that is likely to be a player here.
So, before you go on to the next one, can I ask in that connection, you know, there's a lot of questions here relate to anti-ROS as a kind of a general term, and it puts me in mind of all the antioxidant trials that have been performed, you know, and if anything, the antioxidant therapies are in the setting of ischemic heart disease, and it's probably harmful. So, yeah. How do you reconcile the kind of paradigm that you've presented today with like studies where there's been tens of thousands of patients, many of whom have died early by putting on these antioxidant interventions?
So that's a great question, Phil. First of all, I think I think you can't just target one oxidant species, nor use one antioxidant. I think all the vitamin trials have been, as you said, uniformly negative. They work great in animals at very, very high doses. They do dismally in humans. And so that was one of the strategies and one of the ideas we had from going more upstream and looking at NRF2 to get a multiplex of antioxidant trials. I don't know, antioxidant protein expression. I don't know of any human trials where more than one or two antioxidant vitamins, for instance, or low doses of SOD were tried. Whether or not these proteins get to the areas of injury is another area. You can see that if you give SOD, it's metabolized very quickly. You'd almost need a constant infusion of it. And whether that's the only antioxidant to target is very clear. So I think it's true, and I think that that's one of the reasons we looked at NRF2. Some of the clinical trials that are being done now to study skeletal-based mitochondrial disorders, for instance, are using a compound called RTA408, which is a very potent NRF2 activator, and dimethyl fumarate, and these seem to work very well. Whether they're completely through the antioxidant mechanisms or by inhibiting inflammatory mechanisms is not 100% clear.
Okay.
I hope that somehow addresses that. So the next question by Thomas, and I'm sorry, I'm never going to be able to pronounce your name, Thomas, but could you comment on whether ANP and BNP influence sympathetic nerve activity in RVLM area of the brain, and if they exert effects on NRF2? So I think the answer to the first part of the question is yes. ANP and BNP are sympatho-inhibitory. There have not been many studies specifically looking at the RVLM, but whether or not they target NRF2 in their mechanism of action, completely unknown. Certainly would be interesting to find out. Meow Chu says, do you think NRF2 might compensate for the activation of antioxidant genes when NRF1 may compensate. I don't know. I somehow doubt it. It certainly doesn't look like that from our studies. There are still major deficits in antioxidant proteins, increases in oxidative stress, and we haven't really looked at NRF1 very, very closely. So I guess I just don't know. Rosanna Bassani, is there any change in lifespan and or cardiovascular? No, yeah, that's a very good question. So lifespan we haven't looked at, and some people like to call it health span. So in the human trials, Karen Hamilton from Colorado State has looked at NRF2 activation in a variety of degenerative diseases, right? Doesn't pay to live a long time if you don't have a good quality of life. And that's one of the marketing strategies for Protandim is increasing lifespan, health span rather than lifespan. And as I mentioned earlier, we don't see in our models, we don't see a marked improvement in cardiac function. And I really believe any benefit of antioxidant therapy or NRF2 activation is going to be more pronounced in the peripheral circulation than on the heart itself. You get to a point, especially in late heart failure, maybe if you could intervene earlier, it would mean something, but in late heart failure, you get to a point of diminishing returns, and I think it's very difficult to reverse things. Let's see. Another question by Rosanna. One more question. Have you checked how ROS levels in the medulla of heart failure animals treated with beta blockers? So beta blockers definitely reduce a sympathetic outflow to the heart. Beta-1 blockers reduce sympathetic outflow to the heart, basically at the postsynaptic mechanism. There is data showing that chronic propranolol treatment does have a central effect, and there may be a sympatho-inhibition, but that may not be necessarily beta-1 mechanism. It may be a beta-2 mechanism, and I think that there's little data to show that beta-1 agonists like low pressor and those drugs directly affect sympathetic outflow. They may do it indirectly through improvement of cardiac function. The next question from Dr. Rajasthan, how are you? Who's worked in this field for a long time and has published some really seminal work. Is NRF2 always detected at 110 on immunoblot? So yes, correct. So we are looking at the probably at the ubiquitinated and glycosylated translational form of NRF2. As you know, the open reading frame predicts a molecular weight of about 58 to 60 kilodaltons. In our hands, we get much better immunoblotting with the 110, and we've used both polyclonal and monoclonal antibodies. We do see a band also at 58, but the modulation in NRF2 is much more pronounced when we look at 110. And I think most of the community, if you know the paper by Donna Wang, who showed, I think it's Donna Wang, who showed that 110 kilodalton band is probably the best one to look at. I hope, and there may be some disagreement in the community about that. And by the way, the specific antibody was acquired from Abcam, and we can send you the details on that if you email me. Dan Johnson, do you know how alterations in sympathetics via NRF2 may also affect the parasympathetic, for example, vagal tone? That's a good question. I think vagal tone may be modulated if cardiac function increases. We haven't specifically looked at vagal tone. We've done a few power spectral analysis programming, which we've divided low frequency and high frequency components of heart rate power spectral density. And we don't see a whole lot on vagal tone. Of course, we haven't measured that directly. Most of the modulation is on the sympathetic side. Konstantina Statopoulou, thank you. Nice. Do you know if the type of heart failure, HFpEF or HFrEF, affects ROS production in the brain? The answer is, I don't know. All of our models are HFrEF, but it doesn't mean that there isn't a component of HFpEF also. Our rabbit model may have a little bit more diastolic dysfunction, but I don't know. I mean, that's a good study if we had a really good, reliable animal model for HFpEF.
But sorry if I interrupt. I mean, do you think there is a good model of HFpEF at all? In your opinion?
I don't know. Unequivocally, some people believe hypertension is a model. If you raise pressure, you get to a point where you have diastolic dysfunction before you have systolic dysfunction. In our pacing animals, if we look at them early on, they get cardiac dilation at 1 week, their ejection falls from 70 to 50, 55 perhaps, and it looks like they have some diastolic dysfunction. So I think that may be a model some people are starting to look at more closely. But I don't.
An early stage of HFrEF.
I don't think we know what HFpEF is. I think the problem is that it's likely to be a combination of a hemodynamic and a metabolic disease, and I think that's something that I think we'll know soon. But there are several people that are making, I think, headway in this case. Sinha Hadapur, how does NRF2 become work in the first place? So, that's an interesting question because we did some work on exercise training. And when we exercise train animals with heart failure, I always find a decrease in oxidative stress in the brain. And I said, how does that, I don't understand how it works. How does something from the periphery, from changes in blood flow to skeletal muscle, to changes in shear structure, relate into changes in oxidative stress in the middle of the brain or in the brain stem? And I attended a symposium at EB chaired by Karen Hamilton and Joe McCord several years ago, and they talked about NRF2, and kind of a light bulb went off, and I said, gee, this is something we have to look at. And so that took us down this road of looking at the role of NRF2 in heart failure. And as I mentioned, we looked at this in exercise training as well, and there's no time to talk about that, but it has an interesting effect. Have you considered expressing NRF2 with mutated expression site to allow for more long-term and stable expression? Is it possible that the discordance between, we've thought of that, we haven't done it. Ubiquitination is, as the term would imply, very ubiquitous, and it involves the turnover of many, many proteins. We didn't think we could be too discrete. That's why we took the route of focusing on Keep1, which may also be involved with many other proteins, and using viral transfection. But if we can do that, I think it would provide additional evidence. Farhan Rizvi, what media do you use for fibroblasts to get exosomes, and how much media was collected for exosomes, and what media was serum free? So Changhai Tian, who's listening in, I think who did most of that work, can provide those details. And if he just types in the chat window, I'll go there and I'll provide you that when we're done with the Q&A. Have you tried other means to manipulate ROS centrally and analyze cardiac phenotype? We've used angiotensin, we've used TNF alpha. Those are the main mediators we use to upregulate ROS centrally. Certainly, we didn't find anything else that really did it as well. Angiotensin is great. Other types of inflammatory mediators work very well as well. How exactly, so Jadia Cruz asks, how exactly those cardiac derived vesicles reach the brain and in those specific regions? So I don't doubt that exosomes get every place in the brainstem. I think that they may get every place, even in the cortex, and I'm sure that it's a global effect. How they get from the vascular side into the neuronal side is not 100% clear. They're small enough to get through the blood-brain barrier. They get endocytosed, I believe, into neurons. They may be in glia, although we haven't specifically looked as glia as well. There may be specific extracellular markers, such as beta integrins, that are highly expressed in the brain and also expressed on the surface of exosomes and other microvesicles. And if that is true, and we're looking at that now, that would mean that there would be preferential targeting of the brain. But I'm sure they get into other parts of the brain. We just didn't see our labeled tissues, labeled exosomes in other tissues. And it may be we have to look even earlier, maybe in 1 hour or 2 hours, to make it clear very quickly. Let's see. Okay, back to Alabama. Dr. Rajasthan says, is NRF2 always detect? Oh, that's the same.
I think that's the same one.
Okay. Camilla, hi Camilla, from the University of Toledo. Curious about the increase in blood pressure in the NRF2 knockout. Did you notice differences? Did you notice the differences were more pronounced in diastolic and systolic? Good question. No, they're increased both, and we did not see, actually in that study, we didn't look at water intake. And then maybe we need to finish up now, and let me see if Tian wrote back about looking in the chat section. He hasn't wrote back by concerning the media, but if you want to email me, I'll provide you with all the detail, and I'm sure that's in our papers.
By the way, Irv, you can run as long as you want, by the way. There's no time limit here. It's completely up to you.
Let's see if there's another question that I missed. I think we've covered...
no, I think you missed.
Oh, here's some. Can you say whether a NRF2 inhibitors might be one of the future approaches to cancer chemotherapy? Yeah, so it's been shown that activation of NRF2 actually can be deleterious in response to cancer chemotherapy. Because in cancer chemotherapy, you want high levels of oxidants to mediate cell death. And NRF2 inhibitors may be beneficial. I think this is being looked at. I think there are clinical trials now. I can't specifically tell you which compounds are being used, but I think that's certainly something that's on the horizon. Sung Park.
By the way, sorry, you've missed a question just about Camilla's, Vibarani.
Vibarani. Did you perform exosomal microRNA profile? Did you find uncharacterized novel microRNA clusters apart from the ones we looked at? Any plan to observe the effect of antioxidant phytoconstituents on microRNA's expression, suppressing oxidative gene therapy? So the answer to the last question is no, we don't have any plan to do that, but I think there are people looking at that in the NRF2 community. We profiled mostly microRNAs that had NRF2 targets and a few others that didn't seem to change. Now we're getting more interested in looking at microRNAs that are cardiac specific that may go then to the brain. There are certainly microRNAs that are incorporated in microvesicular cargo that can go to any tissue. I think it will take a long time for us to completely characterize everything. And so we're trying to be as focused as we can right now. Otherwise, I think it's going to be a little bit difficult for us to finish some of this work. David Patterson. Hello, David. Nice to see you. I hope things are great in Oxford. Does exercise training alter NRF2? So it's a good segue into another talk, but a short answer, yes. In heart failure, exercise training reduces NRF2 in the RVLM, along with sympathetic nerve activity and oxidative stress. Ahmed Wafi who's just finishing this up, has a paper looking at curcumin in exercise, and another paper that was published in the Journal of Applied Physiology about, I don't know, 3 or 4 months ago, showing the same thing. So go take a look at that. Let's see. Single bout of exercise. So Dr. Rajesh Skaran from Alabama says a single bout of exercise. Yes, he published a very nice paper not too long ago. That's a great comment. You should look that up. I think exercise training has a big effect. And the last question: any effect of these microRNAs, integrameres, on selenocysteine transfer RNAs? Good question. I don't know. I just don't know. So
Okay.
with that, I think that's the end of the Q&A.
Well, thanks, Irv. That was very good. I think that you may have missed one or two, but if anybody's question hasn't been answered, they could simply send you an email and you'd be happy to take a look.
Yeah, and I just want to, I forgot the last slide I had, and I'm not going to share my screen again, but the last slide was a plug for AJP, Heart and Circ, to tell you that we have guidelines papers, and we have several calls for papers, one of which is the role of exosomes in extracellular vesicles. Expires August 1st. Just telling everybody.
But Irv, please feel free to share the slide. I mean, there's no issue there.
Let me just quickly bring this back up on the screen. So I don't want to be too crass about this, but so here's our advertisement, and you can see that we have guidelines in cardiovascular research. We have our podcast series, which we've downloaded over 50,000 podcasts so far. We're on Facebook, Twitter, Instagram. We have these current calls for papers: environmental inhalants in cardiovascular disease, exercise and cardiac remodeling in normal and athletic states, and and exosomes and extracellular vesicles in cardiovascular disease. And you've got plenty of time till August 31st to meet this deadline. So I hope everybody will take it seriously and send us papers. Thank you.
Davor, is there anything else to say?
No, I think, I mean, I would like to just thank both of you. Well, I would like to thank Irv in particular, obviously, for amazing webinar, great data, disclosing unpublished data, which is quite fascinating. I look forward to the follow-up. And of course, thanks to Phil for chairing, for bringing your expertise to the table here as well. And thanks to the attendees. I mean, we get great numbers day in, day out. And I think clearly there's a need for these things. And the questions and answer sessions are even better, I think. So, so thank you both, and thank you to the audience. And we take a break tomorrow, and then we come back on Thursday with Dr. Tim O'Connell.
Thanks. I enjoyed it very much. Thank you very much.
Thanks again.